Okay, so I want to welcome all of you to a very special event. Today, our guest speaker is Bruce Tromberg. He doesn't need much introduction, but in case you don't know Bruce well, he graduated from Vanderbilt University before coming to the Beckman Laser Institute at UCI. He did a postdoc under Bo Den at Oak Ridge National Laboratory. He's was assistant professor here at UCI from 1990. He took the helm here at Beckman as first the interim director and then the director for about 15 years, from 2003 to 2018. Bruce, while he was here at UCI, he was one of the leaders in the Chow Comprehensive Cancer Center for the oncoimaging program. He was also the director of the laser microbeam medical program. In 2019, he accepted the director position for the National Institute of Biomedical Imaging and Bioengineering at the NIH. And today he's going to tell us about a lot of the work going on in Washington with responding to COVID-19. So with no further ado, welcome, Bruce.
Thank you for having me. It's just really wonderful to be back and have the opportunity to give this talk. I think, as probably most of you can guess, I would much rather be there right now. We're about to have 8 inches of snow tomorrow. This is what they tell us. And in addition to the pretty exciting weather, we've got some climate change coming up, as in political climate, which is also very exciting, keeping everyone challenged. But it's really wonderful to have the chance to be able to talk. And thank you so much, Richard, for reaching out and scheduling this event. The whole Applied Innovation team. I have a fair amount of ground to cover, and hopefully this will stimulate lots of discussion. This is, of course, not how any of us had anticipated we would be spending our time. And I just sort of like to remind everyone of the time when we were all together without masks. So this is a talk that I'm presenting on behalf of the entire NIBIB, a photo just before we went into isolation, quarantine, and reminds us of what we're aiming for and hoping to achieve, and hopefully we'll arrive within the next few months with so much optimistic news about vaccines and expansion of testing. So today I'll talk about the rapid acceleration of diagnostics, and it's something that I guess my entire career at UCI and all of my training had prepared me for, but I didn't realize it until we were put into this position. And the position is one that my infectious disease colleagues, you've been hearing from Tony Fauci quite a lot, and he's been thinking about this his entire career. And I just hadn't ever thought about this as a serious threat, although Tony fortunately had been building the Vaccine Technology Institute, which has facilitated the development of vaccines very quickly and has helped drive the national response. And from a bioengineering point of view, we at NIBIB are a relatively new institute, just established and really stood up in 2002. So our community, of course, has been expanding, and we've expanded tremendously across the UC system. And we had an opportunity to really think about what we could do in order to contribute to mitigation of the effects of the pandemic. And where we've invested our efforts are really in 3 primary areas: imaging and artificial intelligence, and here it's primarily CT, radiologic imaging, thoracic imaging with AI-based approaches. And we've just launched a national network called MIDRIC, which is a consortium of all the imaging professional societies, more than 20 academic institutions, to really advance this and create shareable imaging databases that will be used and hopefully will accelerate the development of prognostic, diagnostic, and therapy guiding algorithms. We've also invested heavily in digital health platforms. I won't have a chance to talk about either of those 2, although I'll touch a little bit on some of the digital health. And finally, diagnostic test technologies. And that's where the entire talk really will be focused. So these efforts have been supported by entirely new resources, quite unexpected for us at the NIBIB. The fourth congressional supplement involved a $1.5 billion additional appropriation to NIH, and that happened on April 24th. 500 million of that went directly to NIBIB, and just as a helping with scale, our annual budget is around 400 million. So immediately we had an appropriation that substantially exceeded our annual budget, and we created a program called RADx, Rapid Acceleration of Diagnostics, which has taken multiple forms at the NIH, and it's actually led by Francis Collins, the NIH director, and we have 4 different types of RADx programs. 2 of them, RADx Tech and RADx ATP, Advanced Technology Platforms, are entirely focused on technology development, the development of new testing approaches, scaling up, and something that we don't traditionally do at the NIH, which is not only drive innovation, but move it all the way into manufacturing, expansion, and delivery and dissemination. And I'll talk about the processes and some of the products of all of that. So I lead the RADx Tech and ATP program. I co-lead that with my deputy director at NIBIB, Jill Heemskirk. And one of the things that's made this very successful is we've been able to partner very quickly across the government, most notably with BARDA. And BARDA likes what we're doing so much, they've actually invested, they received $1 billion in that supplement, and they have invested $300 million in their partnership with us. So all told, NIBIB is managing a little over $1 billion in investments in new diagnostic technologies. We work daily, have a daily cadence with the Assistant Secretary of Health, the FDA, Department of Defense, CMS, and CDC, and it really takes this transgovernmental effort to make all this stuff happen. One thing I'd like to just re-emphasize is that this appropriation came on April 24th, and we launched the program on April 29th. So that's our first example of unprecedented speed, and we'll be talking about scale next. The intent of the program and why everyone was so supportive and we were able to launch it so quickly was to expand testing technologies, the number of tests, the type, and their access, and optimize their performance, both the technologic performance as well as their operational performance. And I'll explain a little bit in what that means in more detail. But the goal is to have tests that match community needs. If you think back to the end of April, when we were doing, as a nation, about 250,000 tests per day, tests were really only available in laboratories. They were only for people who were symptomatic, and they didn't meet the needs, the diverse needs of the population across the country. There was essentially no testing for screening or surveillance or looking at asymptomatic people who actually constitute the bulk of the spread, the transmission of the virus. So the overarching goal was to move our testing capacity up to about, I just picked a number, 6 million tests per day by the end of the year as a nation. And that's been our target. And we actually, I'll talk a little bit more about how we're almost there. And the innovation that we're investing in and driving has a diversity of risk, both from scaling up existing technologies, which of course is lower risk, to investing in novel technologies. The kinds of technologies that many of you are familiar with, that you've come accustomed to, that are in laboratories, in bioengineering and technology development groups all around the country, but hadn't really emerged fully as a commercial driving force. So what we had hoped to accomplish is sort of shift the balance from primarily laboratory tests to primarily point-of-care tests. And we fortunately had already an existing network, an asset in a sense, to build this entire vision on. And that's the Point-of-Care Technologies Research Network, or POCTERN. And that was established in 2007 by Rod Pettigrew, the founding director of the NIBIB. And I had just renewed it when I first arrived in January 2019. And normally this network, which is a consortium of multiple universities, is producing point-of-care technologies. And typically their track record has been the development of technologies in roughly 5 to 10 years, which is normal for all of you who are familiar with the commercialization cycle. What we did was we hijacked the network in a sense. We met with them. We said, we're about to get this congressional appropriation, and we need to leverage an existing structure in order to dramatically expand the testing capacity of the country. So we beefed up the network and brought in more than 1,000 experts, both paid experts as well as volunteers from all around the country. And some of you who are listening may be part of this network, have interacted with it. Let me just mention a little bit of the components that are in it. So first, it's organized. The coordinating center is at CIMMIT, which is at Mass General Hospital. And then there are 4 nodes, universities, that are key participants and are players in the network: Johns Hopkins, UMass, Northwestern, and Georgia Tech, Emory. And each has unique expertise and a functional role in the network that's slightly different, but our network works together. We also added VentureWell and Biocom, which are nonprofits that we've worked with for quite some time in order to help facilitate the rapid dissemination of resources, as well as provide unique expertise that they have in commercialization. And then we created within this network 3 important cores in order to evaluate and advance technologies. First, a validation core, which is at Emory, Georgia Tech. And I guess I need to update the slide. They've completed about 50 projects. Every technology that comes into the network is evaluated by this independent validation core. And more than 2,000 participants as patients actually have had testing technologies evaluated using clinical studies. Then there's a clinical studies core, which I'll talk a little bit more about. That's at UMass. And this is a way for us to do standard trial design and have single IRB protocols and rapidly advance clinical studies, which, as you know, can take for multi-center types of studies or clinical studies on the order of years. And we've been able to launch these in a matter of weeks to months. And then finally, there's a deployment core, which is focused on issues of supply chain, manufacturing, and linking together with our user community and developing end-to-end solutions that are necessary as you piece together all the components of all these technologies. So the overall picture is to help create new methods and processes for the review of concepts, administering funding quickly. If any of you have received funding from the NIH, you know it hasn't been fast. It usually takes at least a year, even when you do extremely well with your scores just through all the administrative stuff. We have been able to get funding to groups with manufacturing and scale up in a matter of weeks and months. And it also provides expertise and, of course, as I mentioned, testing of these technologies. So in order to really drill down a little bit more and reduce all this to practice, we've built all of our processes around what I think many of you are familiar with, which is an innovation funnel. Our innovation funnel is really no different from others that are taught and employed in commercialization and tech programs all around the country, with the exception of the scale, perhaps. And we've tried to introduce within this funnel a series of gates and processes that would optimize outcome and get the best technologies to come through. So let me just briefly run through what the funnel looks like. As soon as we announced on April 29th the existence of the program, we had, I think, 400 applications started in the first 24 hours. We ended up with about 3,000 applications started. 716 ended up being completed applications, and you could submit your application through that coordinating center at CIMMIT. And this was a rolling submission process. Then each application was evaluated and potentially invited into phase 0 or a deep dive, also popularized by Senators Alexander and Blunt as the Shark Tank phase, because as we explained to them the concept which ultimately led to the appropriation, they really latched on to that Shark Tank-like idea. So that's a week-long process where each project is teamed up with a panel, a group of up to a half a dozen experts in every aspect of the technology, the clinical component, the manufacturing, the distribution, commercialization side, and every aspect of the technology is carefully evaluated, and a plan is put together, customized for that technology to potentially go forward with a series of initial investments which would allow us to validate and de-risk the technology. And that's phase one. So of the 136 that went through that deep dive process, 47 made it into phase one. So you can see we're already having a huge step down pressure from that 716 full applications. And I should point out, you can see it on the slide, 30% of these applications were from California institutions, companies, and so forth. That's the overwhelming majority. 47 went into phase one. These awards are typically about up to $1 million and roughly 6 weeks of performance. Then those who make it through phase one are eligible to move to phase two, which is the manufacturing expansion phase. And between RADx Tech and RADx ATP, we've now publicly announced 22 companies in that manufacturing expansion phase have spent about $480 million on that manufacturing expansion. And just over the past 2 weeks, we've moved several more companies into that phase. Probably once we're done, which will be within the next couple of months, I'm anticipating approximately 30. But you can see that's only roughly 3 or 4% of the total number of applications that come in to the innovation funnel that will make it to that manufacturing expansion phase. Typical awards there could be in the $20 to $30 million ballpark. And again, and just to really underscore this, normally it takes 5 to 6 years to make it from your idea all the way out to that manufacturing expansion and distributing tens or hundreds of thousands of tests per day. And we've tried to compress all of this into a 5 to 6 month process. And to summarize who's made it, 17 of these are nucleic acid based testing technologies, 5 are viral antigen. The settings are roughly evenly distributed between point of care and laboratory. From a regulatory point of view, we work in partnership with the FDA. So every one of your steps for validation and clinical testing has baked into it all that is necessary in order to meet the rubric or the templates for emergency use authorization. So if you make it through the funnel, you will have emergency use authorization. And in terms of impact, we're projecting of our investments that have been disclosed, the 22, they'll be generating about 1.5 million tests per day in January, but we still have more in the pipeline. And there are about 20 that are still in process. More than 1 million lateral flow assays will be coming out of that pipeline process in the first quarter of 2021, more than 1 million tests per day. And with other manufacturers expanding all around the country, actually all around the world, we'll be seeing tens of millions of lateral flow assays that come out and that are widely accessible for home use within the next few months. And I'll talk a little bit more about that as the talk progresses. So the idea behind our investments is really to stimulate innovation, and it's been very exciting to see innovation in all of the steps of these tests. Separation and concentration. In fact, one of the performers, one of the companies, and these are all the companies that we've invested in on the right, Series Nanosciences, their entire platform is based on a concentration step that actually concentrates the analyte by up to an order of magnitude, improves the limit of detection by anywhere between a factor of 2 and 10. Microfluidics, of course, have really made the big time with all of these diagnostics. There's so many that rely on novel microfluidics components. Chemistries like CRISPR, for example, one of the companies is Mammoth Biosciences. Next-gen sequencing, those have also been ones we've invested in, though we may be a little bit too soon for next-gen sequencing to be cost-effective. Time will tell with that one. Novel labels and reporters, really interesting readout technology, miniaturization, and of course, automation. And where were we? By the end of November, RADx supported companies were, this is not just theoretical, 600,000 tests per day from RADx companies were being sold. And we actually even had more capacity than that. And that's one of the continuing ongoing efforts, is to make sure that all of the capacity that's generated by these tests is actually utilized by people all around the country. This is a kind of the eye candy snapshot, so you can see what some of these tests look like. In the upper left-hand corner, you can see a handheld Rt-Pcr device made by Mesa. And well, you can see it even more in a real sort of size, tangible way here, holding in my hand. This platform is around $300 or so, and there's a cartridge that goes into it. The cartridge is replaceable, so you can purchase a new cartridge every time you do a test. It takes about 30 minutes. The limit of detection, the sensitivity of this is pretty close to laboratory. It's about 1,000 copies per mil. It's been validated in our core. You can see another one of these made by Visby Medical, which actually has no insertable cartridge. It's the entire device itself is a disposable test. And it's also a California company. There are quite a lot of California companies here. Quidel makes the Sophia platform. It's a viral antigen test. It uses an electronic reader and a lateral flow assay format. You may be familiar, and people have heard about dipstick assays. We're certainly supporting those. Those are lateral flow assays. One that's quite notable, I just want to pause for a moment on, is Illum. And this is an assay. It's a point of care lateral flow assay. It's a viral antigen assay. And it's got their key innovation is this cartridge. And inside the cartridge is really sophisticated disposable optoelectronic components, quantum dots, a Bluetooth chip. It communicates with your phone. And kind of the really cool thing about this is that here we are with Illum that came in with their initial concept 216 days ago, and they just got emergency use authorization for home use over the counter, both for symptomatic and asymptomatic. Really excellent performance with that lateral flow assay. And it uses quantum dots and advanced technologies that have been in laboratories. Of course, Illum has been working on these for some time, but our ability to support them both with resources, dollars, as well as expertise, has helped accelerate the growth of this. And hopefully, we'll be able to have access to these in homes at scale in the coming weeks and months. So not all the technologies can make it, of course. And I mentioned of the over 700 applications, only around 3 or 4% have made it to the manufacturing expansion stage. But many of these are incredible ideas, and we don't want to see these drop. And we've been able to leverage our seed network at the NIH, which is a consortium of centers that includes REACH centers, CAI, the Center for Accelerated Innovation across the EC system, and other accelerators across the country. Most notably here, you can see ULP, University Lab Partners, is now a member of this RADx Next, we like to call it, or redirect program, where projects continue to receive some funding, and importantly, continued industry coaching and mentoring, training and resources, so that these early stage projects, there are about 50 of them that are distributed in this network across the country, are going to continue to progress, we hope, receive some private investment. And many of these are the real transformational technologies that will make a difference in the next pandemic, that will have an impact in other infectious diseases and in other diseases, in cancer, heart disease, and diabetes, and so forth. So I'd like to just take a few minutes to talk about our core facilities, because this is a really unique feature of the network and one of the, I think, essential components to helping us understand which technologies to invest in. So this validation core first assesses the feasibility of the technology itself, and then looks at contrived samples in a variety of forms, and then patient samples. And we get a very detailed report And this report, for me, is kind of like the Sunday New York Times. It's essential reading, and it's really impossible to make a decision without this very comprehensive analysis. And part of the beauty of this is that the validation core we can leverage to compare technologies within a class, so different viral antigen tests, but also be able to compare performance across different platforms, a viral antigen test versus, for example, a nucleic acid test. And this has been a major point of confusion all around the country, actually all around the world. People are not sure which test to use and when, and why should they use one versus the other. Viral antigen tests are much less expensive and faster. So clearly, if they perform just as well as nucleic acid tests, why not use those all the time? And the answer really is in what we've been understanding and learning about viral loads all across the population in people with COVID who are both symptomatic and asymptomatic. And we see an incredibly wide range, around 8 orders of magnitude, from about 10 to 10 to the 9th copies of RNA per mil. Now, the viral loads are generally higher with progressing time after onset of the disease, but unfortunately, and this is a confusing part, the viral loads are not necessarily correlated with infectiousness. And this is particularly true after you've hit your peak and you may be shedding virus. And if you've had pretty bad symptoms, it may take a longer time for you to shed virus and be on that tail versus being asymptomatic. But you may not actually be infectious at that time. So this has made it very hard to figure out how to do different tests. And just you can visualize the difference between a lateral flow assay test, which typically has a limit of detection of around 1 million copies per mil. Its sensitivity, if you look at what fraction of those viral load distributions across the population it captures, is going to necessarily be low. It can be as low, and many are reporting as low as 40% versus RT-PCR if you're looking at all symptomatic and asymptomatic. But if you look at the sensitivity and specificity on label for what many of these are EUA approved for, then it can be well into the 90s for that symptomatic population. The EUAs are typically issued for individuals who are within 5 to 7 days of the onset of symptoms. So this is a very important way to understand when is the right time and the right procedure to deploy in using a lateral flow assay. If we compare this to nucleic acid sensing technologies, their limits of detection could be for laboratories as low as 100 copies per mil. So it captures that whole wide range of viral load all across the population, and that's why they're performing better. But it's really probably not fair to compare lateral flow assays to RT-PCRs every time and very explicitly, because the lateral flow assays are more sensitive to infectiousness. You may not be infectious at those very, very low early stages with low viral loads. So this gives us guidance that we should use lateral flow assays on label within 5 to 7 days of symptoms. They perform extremely well under those conditions of elevated viral load. Off label, their public health authorities, the CDC, recommend having backup PCRs, for example, in regions where there is very, very low prevalence because you may have a false positive. And if you've been recently exposed and you have a negative, again, it's important to have a backup PCR. But another interesting feature that's brought up by Michael Mina and many of his colleagues is thinking about the time dynamics of the infectious or of the viral load expansion in each person. So if you can imagine doing 2 or 3 lateral flow assays in sequence, you may miss the appearance of disease early on, but if you do 2 or 3, you will time it with that expansion of the virus in that person and then be able to have a true positive measurement and have that then nicely correlate with infectiousness. The clinical studies core is really unique in that we have a standard trial design with master protocols, and these studies are typically powered for around 200 subjects. We also use a digital health platform developed by Eureka at UC San Francisco, And this is a wonderful way to do patient enrollment, recruitment, symptom tracking, and technology use results. And there's a single data safety board in IRB, so we can rapidly implement and report the results of these trials. And UMass also has access to a hub of networks across the country through our CTSAs. And so far, where we are, 2 technologies have been thoroughly tested with hundreds of subjects in this core. 3 are in progress with approximately 1,000 subjects that are going to be enrolled. And the Quidel study is a multi-site study at UMass, University of Illinois, Urbana-Champaign, which is also one of our RADx performers, and Johns Hopkins, or RADx sites in Johns Hopkins, where there's this test is being explored for the power of being able to do sequential lateral flow assays, and individuals are being followed for up to 2 weeks, along with RT-PCR tests from both nasal swabs and saliva, and viral infectiousness assays to get to that critical question of how well does a lateral flow assay really predict and tell you about infectiousness, and can multiple lateral flow assays take the place of more expensive PCRs. And we're also in the process of looking at lateral flow assays in a home testing setting and trying to understand self-sampling and the value of digital health platforms and whether they can truly break the chain of transmission. And the last core that I'd like to tell you about is the deployment core. This is kind of our interface between the RADx program at NIH and all of the rest of the country, states, schools, universities, and so forth, businesses. And it occurs through bridges that we've built between NIH, universities, nonprofit foundations, and also industry. And one of the things I'd like to highlight here is whentotest.org, which is a website, and this is a calculator. It's actually a very nice computational model developed by Annette Hossoy and Paul Tessier at MIT and MGH. And the really interesting thing about this, it provides a lot of practical advice and guidance to organizations about how they could stand up a testing program and how much it will cost. So for example, you can enter the number of people in your organization, and also have a variable entry with respect to mask wearing. Even if you go more deeply into the layers, you can specify different types of masks with different performance. It takes into account contact tracing and the type of activities that are ongoing in your organization. And the default is sort of a typical prevalence, but you can also ask it to calculate in the presence of a hotspot or change the prevalence of disease in your region. And you can then see for different testing options, and the default options are different types of antigen tests, a PCR test that would be at point of care, and a PCR test that would be in the laboratory, how much testing will cost your organization per week, and how many times does each individual need to be tested, or how many tests do you need to perform per week for your organization. So it's a really easy to interact with, but as you peel back the layers, it's got lots of sophistication, and it's all designed to minimize transmission within your organization to reduce community spread. So I'd kind of like to wrap things up or conclude with a few thoughts. We've really stimulated and are seeing a shift in balance between laboratory tests now and an expansion of point of care. And in general, we have 2 wide choices of point of care. We've got point of care RT-PCR or PCR techniques that may not use RT processes. We have point of care antigen tests, some with readers, and some will just be really inexpensive visual readouts in dipstick formats. And their cost varies. And interestingly, the RT-PCRs are not wildly more expensive than some of these inexpensive assays. For example, as a point of reference, I mentioned the Mesa Acula unit itself is only about $300. Others may be several $1,000s, but we're seeing the biggest differences between these platforms are in terms of limits of detection. So the PCRs have limits of detection that are maybe 1,000 copies per mil or less, and the lateral flow assays are much higher, maybe 100,000 to 1 million copies per mil and greater. And that's where we need to be able to bridge the gap. And there are a number of really exciting technologies that are under development that will come. Many of them are in that 50 group that are in the redirects that will truly bridge that gap. But there are also some that do exist, and some involve using our emerging technologies a little bit differently. And one of those is the concept to pool samples together, and pooling a point of care PCR technology in a small social network is an approach that many are exploring, including professional sports leagues. But I think it would be really quite applicable for families, co-workers, communities who may be interacting on a regular basis, who, if one of them received individually a positive test, others would have to go into quarantine and get a test anyway. There's a slight reduction in performance with these because of a dilution effect, but there's very fast turnaround. You get the answer instantaneously. The cost is reduced substantially if you pool 3 to 5 people in a single assay. So if you take this cartridge from Mesa, the cost per person would be comparable, $5 or so, to a single point of care lateral flow assay test, but the performance is better. And the answer that you get will be fast, and you can isolate the entire social pod instantaneously and reduce the spread transmission. The last piece of the puzzle, and I alluded to this right at the beginning, is how do we integrate all of this into information that can be used both for individuals and for public health? And this is a fast-moving area, and we'll see more expansion here. Many people have strong opinions about it one way or the other, as in we must have digital health capture. The value is the actual output of the test can be read directly by readers, optical readers, that can look at lateral flow assays, for example, and interpret them even more with a higher level of performance than your own eyes. You can integrate instructions that are much easier to follow than folding out a giant instruction sheet and trying to follow that from each manufacturer. They can follow symptom surveys, and this is one of the apps that we've supported the development of, and we're actually using at the NIH for assessing symptoms every day for every employee and understanding who can go back to work and when. There are opportunities for privacy-preserving digital contact tracing if you get your information in there. Some states have already signed up for this Google Apple notification service. It's currently in the iOS. If you have signed up, all you have to do is press a button if you've tested positive. But clearly, if it's integrated with your testing input device and it becomes automatic and also links up to electronic health records, then potentially you can get a kind of passport, a digital passport. And so that's the ultimate incarnation of it. We've been working with the Gates Foundation, the National Cancer Institute, to help accelerate some of these platforms. So just to sort of summarize, the technology development we thought at the outset was going to be really hard. It's turned out the technology development part is hard, but it's probably way harder to implement all these things. Managing to do screening and surveillance to assess the presence of the absence of disease in asymptomatic populations is something that's never been done before in our country, really only recently in others, but others that don't have the same healthcare model and economic sustainability model that we have. So what we need to do in part to make this happen, to get screening and surveillance is to add to that at-home pathway, both over-the-counter, which we've just today received our first over-the-counter, as I mentioned, from the LAMMP technology, as well as prescription. And some of you have seen the PCR assay from Lucira which is a prescription-based PCR home-based measurement. And then we need to create new structures, economic, regulatory, and reporting to be able to support all this activity and information. From a tech development and expansion point of view, we will be seeing millions of accessible tests per day in the first quarter of the year that will be suitable for home use. And we need to figure out how to use them and make sure that they're efficacious, both for public health and personal guidance. And we're hoping to be able to inform the guidance on some existing technology, as well as some emerging ones that really truly bridge the gap in terms of performance between PCR and lateral flow assays. And finally, we're also supporting a number of studies to help provide that insight. For example, the use of sequential lateral flow assays to prevent infectiousness or to assess infectiousness. And then the big one, which is if you do get a lot of tests at home and they are being deployed, do they really break the chain of transmission when put into the hands of individuals? So that's still an outstanding question, not known. There are a couple of experiments that have been done internationally. Some of you have followed the one at Slovakia and in Liverpool, and the jury is still out. Those are probably not ones that we would necessarily emulate the structure of, but hoping to structure something that makes sense for the US. And the last slide, just a summary thoughts. This has been kind of like the Super Bowl for our field. We've dramatically expanded our budget. We will end up investing in new testing technologies over $1 billion in an incredibly short period of time. And it's helped us implement our vision for engineering the future of health, which is, I think, the last thing that I talked about when I was back at the Cove. It wasn't that long ago, a little over a year ago, and talking about where we anticipated going and helping drive the field. And here we are in a position to be able to implement that. It's helping galvanize our community and I think bring attention to what technology can do and the impact, not just in COVID, in infectious disease, but in prevention of disease, really a new role that technology can have continued expansion in medicine. And clearly, building partnerships and stakeholders is really essential for implementation of all of this. The technology part is relatively easy, but getting this integrated into the social fabric of the country is an entirely different challenge altogether. So I'll stop there, and hopefully we have some time for some questions.
Well, thanks, Bruce. That was a great presentation. And so Tom will join us here and we'll walk through some of these questions and have you answer them. I'll go ahead and kick one off. I have a question here from Steve Blank. How do you see the reimbursements, cost, and access changing from these technologies?
So right now, from what we understand, reimbursements are really only going to be for medical diagnostics, a return of a medical diagnostic to a symptomatic individual. And this is what's making the screening and surveillance part of things extremely challenging. Everyone kind of knows, the intuition is we would like to have our eyes on where the disease is across the population and prevent asymptomatic spread. But currently, the economic structures are not in place to support and sustain that kind of activity.
And I'll just jump in with one more, and then Tom will have you field a few questions. But there have been a number of people asking about the recording and the presentation. So this recording will be posted. I'm going to have to double check, but it should be posted soon, in the next couple of days. Is your presentation available publicly, Bruce? Your PowerPoint or?
I think I can probably, let me just go through the slides to make sure it's okay. But if you can definitely publicly post the presentation, and we can go from there with the slides. But
that's fine. We can talk later, and we can post that presentation or you can edit it and we'll post it. So let me shift over to Tom and have Tom ask a few of the questions.
Yeah, thank you, Bruce. That was an excellent overview of all the activities. First question, do you know how other countries have managed to meet the testing needs in each respective country, like China or maybe Germany?
Yes, and in some ways we wish that we could be like them. You know, they have a more coordinated effort in terms of getting everybody to do things a little bit more uniformly. We are quite diverse. You know, I think the common story that's told and retold is how Korea at the very beginning, some people got together and said, we know some of these technologies work really well. We're going to move them through, get them approved. We're going to deploy them. Everybody's going to get them. You know, this like happened in a matter of days. We have a process in the US, and the FDA has been overwhelmed with applications for emergency use authorization. The last I checked, there were about 200 EUAs that were issued for PCR technologies. There were 7 for lateral flow assay viral antigens. And they have, I don't know, it's an astronomically large number in the pipeline. I think what this highlights, these are not medical devices and medical tests that we need to necessarily be thinking about. It's another pathway that we need to think about, a public health pathway. And it's more along the lines of when you're driving your car and you hear a funny noise or a light goes off that says, you know, it's a warning, take it in to get looked at. We've got 2 stages, and we don't have enough check engine light type of tests. We have, the country is structured for, you know, getting an engine rebuilt or a repair job on some component that's well understood to be broken. So it's kind of the clash of those things. This is going to be an inflection point for us.
Thank you. Another question, will RADx be involved in monitoring some of the early vaccine recipients and be able to evaluate how well the vaccines are working and the duration?
Yeah, that's a really, really good question. I'm hoping that we can get, I'm part of this Operation Warp Speed thing, and that's been absolutely stunning to see the developments there. And I'm hoping that we can get some of the tests out to understand the durability of the response in the population. But fundamentally, we are going to have to be a bridge to the vaccines because, as you know, the vaccines are coming, but we're going to be saying that for many months. And it could be end of 2021 before there's really substantial widespread. Don't quote me on that, but you know, late summer, early fall, late fall, it's going to take a while. So bridging to vaccines is still quite important, and there's still a lot of testing to do. Fundamentally, though, the test itself, of course, we need to couple that with human behavior modification. And that's, for those of us in technology development, that can be a bit of a mystery. So that's where partnership with public health comes in.
Let me jump in, and there was a question to ask, will this rapid approach now be a standard approach for vaccine and treatment development, or will this maybe create some changes, if not the norm for the NIH in the future?
I've been really excited to see in congressional language statements and about RADx being a structure that the NIH should consider continuing. You know, nothing is instructive or definitive, but I think the word is kind of getting out that we can, as a nation, go from idea stage to manufacturing and products, you know, that people can benefit from much more quickly than we have under our sort of normal processes. And so I think it's changed the way that we think, and certainly the same for vaccines. Although I have to say, Tony Fauci has really envisioned this from the beginning, and it's because of maybe 10 years of work already in mRNA vaccines through their Vaccine Research Center that they've been able to find this sort of perfect opportunity to deploy that approach and convert on that. And at the same time, I would say for the technology community, it's only because of people's obsession and development over many years. I just showed you this little device, which uses quantum dots, disposable optoelectronics, and Bluetooth, all embedded in an, in an ASIC. And really 5 years ago, 7 years ago, we wouldn't be in a position as a community to be able to support and sustain the growth and distribution of those kinds of technologies. So I think the fundamental investments in the networks we've been able to leverage quickly. That's a big story that's coming out of this.
So let me just jump in with one other, Thomas, and shift over to you for a couple. But Bruce, you've been around innovation for a number of years, and often a crisis creates opportunities and accelerates innovation, which is what we're seeing now. And so, as you look back on a long career in innovation, is there anything you can draw upon how this is going to change in general, other things within potentially the NIH and innovation in general, at least in the medical area, since you've been pushing innovation through the pipe for a number of years, and now you're in an interesting seat to look at this at a more national level and a more NIH level, et cetera. So is there anything that you can also add to what you already mentioned about vaccines specifically with the NIH and how things might change over the next few years? It certainly has changed the way that people think about the role of technology in having immediate impact on our lives. I would say prior to this, there hasn't been necessarily as widespread an understanding that technology that has not been developed or that doesn't exist could actually be developed in a purpose-driven way to solve hard problems quickly. So I think that that's changed the way people are perceiving it. Virtually every program I hear about that is under development across the NIH now has technology in the language of the program, no matter how fundamental the mission of the research is or directed the program is towards humans. It has become just inseparable from the ideation process, and so that's exciting to see. And what comes about as a result of that, I think, are opportunities for commercialization at scale.
Bruce, when you survey the technologies that have come out of your program versus those around the world, could you comment on similarities and differences?
Yeah, what's really interesting is that of those 3,000 applications that are started, I don't think that there's a technology developer around the world, with a few exceptions, who have not engaged or applied to or communicated with RADx. So we've been a magnet not only for the US, and again, I underscore 30% of the completed applications came from California. So Southern California, particular hotbed for in vitro diagnostics. And we're seeing there's some pretty exciting ones, at least on paper, that we've seen from India. For example, they have a CRISPR assay that looks pretty interesting. But another thing I've learned about technology, I think many of you know this, is some of it is too good to be true. So we've seen a lot of too goods to be true. I've gone down many of those paths, hopefully, in hopefulness. And when having this innovation funnel is a different way to evaluate, you get the deep dive, which of course you're very familiar with at Applied Innovation, the intensive review of something, and you get the full experience of many people, and you make your investment before then. It's kind of like a 6-month venture review and analysis that we're doing.
Thank you. I have an interesting question. As you survey the companies that have come out of the RADx program, in terms of, your really small, scrappy startup versus a big, could you talk a little bit about the scale of the companies in their successes and challenges?
Yeah, that's a great question. I mean, the interesting thing is, so we have had startups, small companies, mid-size, large companies. The overwhelming number are small companies. I would say, and this is a general statement, but the validation core led by Wilbur LAMMP will validate this, and I think they're even publishing a paper on it. In general, the smaller companies like the Mesas and the Illums that are out there have been able to produce stuff with better performance. They're more innovative than the very, very big established companies. They've been able to introduce, and maybe this is not surprising, but they've been able to introduce features quickly that are difference makers. But if you want to invest in something that's going to work and work quickly, then you kind of want to invest in scaling up an existing performer. So we've really tried to balance the two. And the announcement today of Illum, which has been in our process for 216 days, which is, I think, that's actually long on a RADx timescale, but they're obviously moving very fast. That's 216 days from idea submission to EUA for home use. That's pretty fast, and that's a smaller size company. Now they have, to be fair, they've partnered with QIAGEN, which is a really big company. So a lot of times the sweet spot happens when a small company partners with a big company that helps facilitate, growth. But we've also seen times where small startups or academic groups partner with big companies, and the big company is more like the weight around their neck, and they actually never go anywhere. And I won't give any examples of that, but you've probably seen some that have been advertised as having received RADx Tech funding, and you didn't see anything else come out.
So maybe we can wrap up with a couple more questions here. I think you addressed this, but I'm going to ask this question to be sure. What is the ballpark number of tests that may be needed per day in the U.S. to help reach a meaningful control? Is that the 6 million a day, or is that number even larger than that, or another number?
So, yeah, people come at this from various angles of their backgrounds and training. I come at it kind of from an angle of signal processing. So if we have, let's say, what is reported nationally from our testing surveillance program is on the order of about 250,000 new cases per day across the country. And our estimates are roughly that every case that is known, there are probably 3 or 4 or more that are unknown. So there may be over 1 million cases a day distributed all across the country. And it's not a perfect sinusoid. So there are all these little point sources of cases that are coming on and off, but 1 million or 1.25 million per day. We need to get our eyes on that. And how do we fully reconstruct that unknown of the number of cases per day? And so we need to be making measurements per day that are some multiple. And so I picked 6 at the outset as a fairly, perhaps a safe multiple. So 6 million a day. However, I didn't anticipate this kind of secondary expansion of cases that we're now experiencing, and it could be 10 million a day. But I do want to emphasize that this is for my colleagues, the technical group. This is a space-time problem. These on-off rates are not occurring equally in all different parts of the country, and so we need to therefore modulate our testing protocols and the types of testing technologies in proportion to the risk of asymptomatic transmission in regions that we can identify. So some places, some geographies may need lots of tests per day, others less. For those of you who are sitting at home, like me, I don't really need a lateral flow assay test every day. I should probably have one of these, and my wife and I should do one of these a week. We could just pool. So this could be quite affordable for a family, and has a high level of sensitivity. And so the testing the technology and the frequency of its application should be adjusted to the level of risk for that individual and what they need to do with respect to their work and livelihood.
Well, Tom and I would like to thank you for a great presentation today. And for those of you attending online, there are a lot more questions we can't get to as we're out of time. But before we sign off, Bruce, is there anything any thoughts you'd like to close on now that you've been in this most interesting seat to see all this happen? Any last comments?
Oh my gosh. Well, just first thanking everyone. It's just incredible to be back, and it's horribly painful to look at all the names in the chat and all the people that are attending and not have a chance to meet and talk and interact with all of you. Have a glass of wine. You know, speakers are supposed to get a dinner, and you know, I was kind of hoping I could get a dinner, but maybe put that into the future, 6 months, 8 months. So I'm looking forward to coming and visiting and meeting all of you and debriefing and talking more. But please don't hesitate to get in touch. Will you save the chat, Richard, so I can look it over and send me a copy of it and try to get back with all of you. And when you're traveling again, please come visit me in Washington. It's been quite exciting to be kind of having a ringside seat to all of this, and proud to be able to represent the technology community at UCI, California, across the country, in having a role in introducing some practical solutions to the pandemic. So thank you very much.
So again, Bruce, great presentation. We appreciate everything you're doing really for the country in making RADx move fast and move effectively. So we will save, I'm going to ask the AV team to save the chat, the Q&A and the chat for you later. Again, we appreciate your time. We know you're super busy, so thank you so much for doing this. Tom, would you like to close with anything?
No, just second what Richard said. It was a fantastic update. So many people miss you here, and we're all looking forward to when this lifts and we can be face-to-face and in person.
And lastly, I'll just say that
thank you, Richard.
Yeah, thank you, Bruce. I'll just say that the Beckman Laser Institute and Beall Applied Innovation will probably be doing future presentations jointly. So look for those. And so again, thank you everyone for joining us today. We will post the recording, and we will certainly owe you that dinner, Bruce. So thank you very much. Have a great day. Thanks.
Thank you.